Welcome, everybody. It's my pleasure to welcome you today to our seminar series and also to have the pleasure of introducing Dr. Hussin, who I've known for a number of years now. Dr. Hussin actually did her PhD here in Montreal with Dr. Philip Awadala at Saint Justin Hospital. And then she did a postdoctoral fellowship in the United Kingdom with the Wellcome Trust Center and Dr. Peter Donnelly, and has returned to Montreal, where she is assistant professor in the Department of Medicine of the Université de Montréal, and also an investigator at the Institut de Cardiologie, the Montreal Heart Institute, since 2017. And so she holds an FRQS Junior 1 Researcher Award and has all kinds of interesting research projects that are ranging across, how one can use genetic data, genomic data, metabolomic data, questions in population genetics, and questions in cardiovascular health, not surprisingly, since she's at the Heart Institute. And we are really looking forward to her presentation. Welcome, Julie.
Thank you very much. So I'm going to share my screen. And start the presentation. And so
that looks good.
Yes. Yep. Okay, perfect. So thank you very much for the invite to speak to you today. So as Celia just said, in my group, we have a lot of different interests that we kind of put together with this idea that evolution can help with understanding molecular mechanisms, but also combining evolutionary theory with the study of human variability might be able to improve medical practice. So in my group, we investigate the balance of evolutionary forces. So by that, I mean demography, natural selection, recombination that act on genetic diversity in humans to understand disease, in particular cardiovascular disease at the molecular level, and also to identify the most effective and preventive and potentially therapeutic intervention for each individual, taking into account their own variability. And so the methods that we use, so I'm an Evado professor at University of Montreal. And so my group is very focused on data science methods and using them and developing them if need be to study these questions. And so our methodologies are bioinformatics, or, generating pipelines, appropriate pipelines to analyze omic data, statistical and population genetics, and also machine learning. But today I'm going to focus my talk on a specific project that we've been busy doing for the past couple of years that was published in eLife last month. And so that was mainly driven by Isabelle Gamache, who's a PhD student in bioinformatics at Université de Montréal, also supported by Jean-Christophe Grenier for bioinformatics analysis and the data analysis. So the main theme of this presentation is about gene-gene interactions, and you could see that term interaction in that title. And so I just want to make sure we are all on the same page for what this is. So it also commonly termed as epistasis. And it's when two or multiple genes will influence each other in a non-additive ways. So for example, here, imagine that you have gene A that defines dog hair color, light or dark, and gene B that is defining nose color. So we have an interaction here because the dark color is modulated depending on variant of gene B. And in fact, the variant of gene A also changes the nose colors that are available. in this example. So I've been wording this as interaction between genes, but in fact, the epistatic interaction appears between mutations. And so here we can imagine that there was a nucleotide change from C to A in the DNA sequence of gene A, and the individual with at least one A will be of dark color. And the dark color tone will depend on whether or not they have a mutation on gene B. So an important question in human genetics is the extent of this phenomenon as it relates to human traits and disease. And today, most models in statistical genetics of association, for example, between traits, do not consider interactions. And I just want to briefly maybe present the methods that do try to detect epistasis right now. And also, so there's statistical methods that in general tend to conclude in favor of the null hypothesis of no epistasis, unless there is strong evidence in favor. So you can see this quote here: if we can reasonably model the data assuming additivity, we prefer this over requiring epistasis to explain the data. And this is also mainly a statistical argument, because one issue is that given the high dimensionality of genetic data, the multiple testing burden means that very large cohorts are needed to detect even very strong epistatic effects. Another newer approach is machine learning methods that would rely more upon split samples with cross-validation to avoid fitting to noise, and that propose to identify epistatic patterns in high dimensional states. So there's no formal gene by gene model necessarily in these methods. And the general idea is that multi-locus genotype combination will be classified as either high risk or low risk combination for a specific trait. And so this means that that you need to base this on, and it's the case in both set of methods, you need to base this on penetrance, so on the probability of a disease or a trait. And this makes these methods highly dependent on the outcome, the measurement of the outcome, and also on the scale of the outcome. So there's a third way of looking at epistasis and gene-gene interactions, and is to look at coevolution patterns in the data. Coevolution approaches that are based on fitness, and here fitness is in the evolutionary sense, the capacity of an individual to pass its gene. And in this case, we don't necessarily have to rely on a phenotype. So if I go back to the dog cartoon, let's say this dog, the brown dog with the brown nose, has the highest fitness, so meaning that it will have the highest number of offspring. Then in the population, after many generations of this selective pressure, we will see an excess, an overrepresentation of dogs with these genotype combinations compared to the others, also taking into account allele frequencies and so on. So then we will see that this is overrepresented. The problem with this approach is that we won't know necessarily, we will see this, we won't necessarily see that it relates to the actual dark color and nose color. And then we need to investigate what is associated with this combination in order to understand the pressure and the impact of the gene interaction on phenotypes. So when I started at the Montreal Height Institute, and I talked about population genetics and evolution with different researchers. At some point, Marie-Pierre Dubé and Jean-Claude Tardif, who are researchers at the Montreal Hyde Institute as well, they asked me this question: would it be possible to use this idea to potentially investigate the relationship between two genes of importance in pharmacogenomics? And so they had a specific case in mind, and they told me this very, very intriguing story that they had about the pharmacogenomics of a drug that I didn't know about, which is called the dalcetrapib. It's a CETP inhibitor. And for those of you who don't know what CETP is a protein that mediates the exchange of cholesterol esters from HDL, good cholesterol, to LDL. And so the idea of CETP inhibitors is to block this conversion. And so there has been different ones with limited success in clinical studies. And one clinical study of dalcetrapib is DAL outcomes. And it had been shown that dalcetrapib would, yes, lead to an increase of HDL cholesterol levels, but it did not lead to a reduced risk of recurrent cardiovascular events in that trial. So it was stopped for futility, but there was the possibility of doing a pharmacogenomic study. So a pharmacogenomic study is when you look at different genetic variants and you try to see if there is an association between the drug efficacy and some genetic variants. And so what they found was one genetic variant in the ADCY9 gene, which is called, which here is this RS1967309, and I will call it 7309 in the rest of the presentation. And as you can see here in the cumulative incidence curves, is that if you are AA at this loci, at this locus, sorry, you have a reduction in the chance of having a cardiovascular event. If you are AG, you are like in the placebo group, and if you are GG, conversely, you have an increased risk of cardiovascular event. So that was very intriguing, and it led to a second trial, which is called DALGene, to test whether there's an advantage of having the air allele, which would lead to a benefit for the drug. But the questions that Marie-Pierre and Jean-Claude had was that this mutation in ADCY9 was in an intron, and it was really of a known function. So it was not easy to understand what was going on there. And also, and probably more importantly, what is the potential link between ADCY9 and CETP? That wasn't known. So we decided checking with population genetics and data science approaches what the different data sets that were available to us would tell us about these different genes. So the first thing for this coevolution hypothesis is that you need to have positive selection acting on the gene or on the SNP. So the first step was to test whether or not we could see selective signal at 7309 SNP. And so here what you see is a selective a selection statistic, which is called IHS. And if the value is over 2, it means that there's some evidence for selection. And so as you can see within the gene, there's many different potential signals. So it's a gene that is potentially under selection in different human populations, so African, European, East Asian, South Asians, and people from the Americas. And so what we saw at this specific site is that there seemed to be a signal specific to South Asian, but it made it a bit more promising that something was going on in that LD block of the association, and that potentially selection could act on this locus. Then Isabel went and looked more closely at the allele frequencies, and we could see that in Africa, the allele frequencies of the A allele was quite high. The A allele, I remind you, is the one that could be beneficial. But then in Europe, we can see a drop of this allele frequency and genotype frequency. And she also noticed an anomaly, which is that in one of the population of the Americas, there was this very high increase of allele frequency or genotype frequency of the AA, which was not seen in other American populations. So we thought of looking at this or formalizing this a bit, and there's this selection statistic, which now doesn't necessarily look at selection within a group, which was the case for IHS, but now more across groups. And that statistic was developed or presented more than a decade ago and led to the discovery of genes that were involved in high altitude selection for in high altitude. And so the idea is to compare a kind of a tree between three populations. So here they took the Danish population of European descent as an outgroup, and then two Chinese populations, the Tibetan population and the Han Chinese population. And under neutrality, you expect that the largest branch is the Danish population, and then these two branches are quite small because these two populations are very close together. And what they saw is that there was some genes, and in particular APIS1, that was really, really distinct from in the Tibetan population, and that made this branch very, very long compared to the Han Chinese branch. And so we decided to use the same approach in our case in ADCY9 at our SNP. And so again, here you have probably what we expect under neutrality. So this is an outgroup, East Asia. We selected this. We didn't want to have an European population because we know there would be some admixture between Europeans and American populations. And so we expect that Peru and Mexican populations would be quite close here, and that the the East Asian branch would be larger. And this is what you can see from our 99% top 1%, 99 percentile of the length of the branches. So it's quite high or higher for East Asian branches. Then here you have the Peruvian and the Mexican branches that are expected to be smaller. But then if you look specifically at 7309 SNP, you can see that, well, so for the East Asian branch, it's within, it's not in the top 1% of values, but that specific value SNP has a value well above the top 1% value in Peru and not in Mexico. So this confirmed in some sense that there was the potential of a selective signal that had happened, that is acting on this SNP. But this being said, it doesn't really tell us what's going on between ADCY9 and CETP, which are on the same chromosome. By the way, I think I forgot to mention that, at 50 megabase away. And so we set up to understand whether or not there was a genetic association between these two. So to find coevolution in genes, there's different approaches that have been used. One the first method that has been used is to test the correlation between species or gene families. In our case, CETP is not a gene that is necessarily conserved, even across mammals. For example, mouse doesn't have CETP, so it wasn't appropriate method. Another method has been used is to look at the allelic association across populations. And this has been used notably in this paper by Single et al. In 2007 to look at the correlation between HLA and CUR, and was a very important discovery and led to a lot of follow-up research. And here you can see that we see a correlation. So in each population, we compute the allele frequency of the two different loci, and here there was a high correlation, significant for the association between these, suggesting that in fact these allele travel together across evolution among populations of humans. Another way is to look within populations. So for one specific population where we think we have this hypothesis where that there's coevolution, would be to compute linkages equilibrium. So probably most of you know linkages equilibrium as a statistic that we compute for nearby genes, SNPs, to see if they are associated, they are in the same LD block. But in Drosophila research, it has been done a lot and shown that between different genes across the genome, we can compute these statistics, and that there's functionally related genes will have a higher LD than non-related genes. And so that has been also demonstrated that some of these genes are actually interacting. So one statistic to compute LD, famous statistic, just R square. Here you have the definition, is what we went for first screen of the effect, potential effect of potential association between 7309 and other SNPs in CETP. And so what you see here is a plot of all the different SNPs in CETP correlated with 7309. And we saw 3 different SNPs that were above the 99 percentile, suggesting that potentially there was something happening here, but also maybe that they're false positive. So then Isabel made the opposite analysis. So she took each of these 3 loci and then went back to the entire gene of ADCY9. And here the line here represent the top 1% of all the combinations possible. And so what we saw here was this bell-shaped pattern around 7309 for specifically that SNP that I will call 477. And so this was our kind of main lead SNP that we wanted to investigate more to see if we could confirm that this was actually empirically unlikely to have this association, given that these 2 genes were 50 megabase away from each other. So we asked how likely is it empirically? So given, we are here in the, sorry, I forgot to mention this, but we are here in the Peruvian population with, structure, LD structure and specific to this population. So we wanted to know how likely this is. And so Isabel took many pairs of SNPs that matched these 2 SNPs in terms of allele frequencies and distance between the SNPs, so 50 megabase away in the genome. And what we could see is that our pair of SNPs was significantly, the value was significantly higher than the null distribution that we built, according to this null distribution. So adding evidence that this might be unlikely to see. So she went to look at the allele frequency profile for that SNP 477, and strikingly, so we see the same allele frequency or a pattern in Peru that's expected because we just did the correlation. But across the human population, we saw something very similar. And if we formalize this a bit more, you can see that there's a strong correlation between these frequencies across populations. This correlation is higher than what I showed you before with HLA and CUR. And so that was very striking. And what was more striking even was that Peruvian result, which was, yes, correlated, but also outside of the group of, American population. So it seemed like something was really happening between these SNPs in that specific population. Here, I won't be discussing this too much because of time, but we also looked at a data set of Native American populations, and we discovered that in the Andean population, this SNP was already at high frequency, which led to the hypothesis that since Peruvian have this ancestry as well, the Andean population, which led to the hypothesis that this rise in frequency happened during the migration of the Native Americans, and that the Peruvian inherited this effect. And we saw that the association, and this data set has a very low number of individuals, but in that data set, we could see an association as well between 477 and 7309. And this was done by Isabel in collaboration with Oli Trochet in a postdoc in my group. So then, of course, we see this, it's very interesting, but we need to make sure it's not a fluke. There was some lines of evidence that were pointing towards that promising association, but 1,000 genome population is quite small, 100 individuals on average. And so we needed to make sure that this was an effect that could be replicated. And so thanks to a collaboration with researchers in Harvard Medical School, we had access to a Peruvian cohort of over 3,500 individuals. And you can see here that doing exactly the same analysis led us to find the exact same SNP. Then we did the reverse analysis as we did before and saw that the same region was highlighted in that second data set of 3,500 Peruvians. And if we compare the R-score values between the two, we can see that a bunch of SNPs are false positive in the 1,000 genome, also in LIMAA, in the LIMAA cohort, which was that cohort of 3,500 Peruvians. Our first locus were also false positive, as we can see here, but our association was kind of the strongest in both, and that was that confirmed in some sense that there was something happening between these SNPs, or at least between the LD blocks over there. So, yes?
Yeah, do you want to, I didn't ask you, do you want to take questions during, because there's one that's just come into the chat.
Yes, yes, of course. I don't see the chat right now.
It's from
but,
Gravel, and he's asking,
yes.
what's the proportion of indigenous ancestry in your LIMAA cohort?
Yes, so it's quite high. It's the most, in the LIMAA cohort, it's similar to the 1,000 genomes. I don't know if it speaks to him. So we have all these plots. Yes. so we have all these plots that I don't have here, but it's very similar. So there was some groups in the LIMAA cohort that were of higher Indian ancestry, some that were of lower. They clearly, form subgroups that we took out. We investigated each subgroups, but like within, so it's not 300, 500, like final number that is here. But yes, there's lots of details that I can give you if you're interested. But overall, the PEL and the LIMAA that we took were very similar in terms of ancestry.
Thanks.
So the next step also was there's other ways of looking at coevolution, and one other test that can be done is the genotypic association test, which was not feasible in the 1,000 genome population because it was too small, but in that LIMAA cohort, because of the number of individuals we had, it was a lot more, it was feasible to do this. And the reason why we are interested in those other tests is that not all scenarios will be detected with R-square, because there might be some other combination that are not linear in some sense in terms of the allele frequencies. So for example, if you have this scenario where you have an enrichment of these alleles, these genotypes, you wouldn't pick up this association with R-square. And so we went on and we wanted to know more about which combinations were actually enriched. And so we used that chi-square test, where we could have this value of association and also detect which are the groups that are the most enriched, knowing that we have seen with R-square this result, but we wanted to make sure we knew which combination was enriched. And to our surprise, we saw that there were two combinations that were enriched in our data set. So very high chi-square value. In this case, we have to be very careful in terms of the significance, because it depends very highly on the allele frequencies. And so we did permutation tests. So again, empirical p-values, and it was very significant as well. But then we investigated this a bit more. Why do we have, so there was a signal in R-square, mainly driven by this combination. But so why do we see these differences? And after a lot of analysis, Isabel finally understood that, in fact, the correlations and this coevolution, or signal at least, was not the same if we stratified the cohort by sex, with this genotype combination mainly enriched in females, and this genotype combination mainly enriched in males, and which was the one driving our R-square signal. And you can see this here. So if we split both cohort, PEL from 1,000 genome and LIMAA from, and the LIMAA cohort by sex, we see that the signal from the R-square test is mainly and only seen in males, which, as you see, replicates. So then we went on to understand, what could be the phenotypes, and the first step would be to look at the endophenotype that could be driving this, whether there is an actual biological function linking these two genes from these two SNPs. So the first step was to see if there was an effect on the expression between the two genes. So the team of Jean-Claude Tardif, so Eric Reaume and José Sanchez, what they did was that in cell lines, have G2 cell lines, they knocked down ADCY9 and looked at what was happening with CETP. And what you can see is when you knock down ADCY9, there was a significant increase of CETP expression. This was done in an RNA-seq experiment, so it was in the top 1%, like even more, of all genes modulated by this experiment. So it was quite convincing, not direct evidence, of course, but that there was an effect of ADCY9 on the regulation of the expression of CETP. Then we noticed that SNP 477 was reported as an eQTL of CETP in multiple data sets. So we confirmed this in the Jovadis dataset, which is a data set measuring expression of a lymphoblastoid cell line from the 1,000 genome European individuals. So here, I just want to emphasize the fact that we're not looking at a Peruvian data set, we're looking at Europeans. And so the QTL p-value it was quite high in this data set. But then in GTEx tissue, we also found that there was multiple tissues in which that SNP was an eQTL. And so then it led to the hypothesis, or at least the analysis of looking at the interaction between those two SNPs on CETP expression. And so what you are seeing here is the different SNPs, so 477 and 7309, stratified in each case. So here you are seeing this eQTL that I was just reporting. So when individuals are AA, we see an increase of expression with each A allele in 477. But in heterozygote, this effect is attenuated and is not seen at all for the individual that are GG. The interaction p-value is significant and led us to the hypothesis that this was actually an interaction in QTL. We replicated this in the Carthagène cohort, which has RNA-seq from blood. And so here I'm reporting things a bit differently. So I'm reporting this p-value of interaction according to the number of peer factors. So peer factors are a way of capturing potential hidden factors that are biasing gene expression measurement genome-wide. And it's never very clear how many you should use in your regression models. So we plotted them all to, the limit of getting enough power. But so what you can see is that for most number of peers, we have a significant effect, interaction effect of the two SNPs, so 7309 and 477 on CETP gene expression. Given the hypothesis or the observation about sex-specific effect, we stratified the cohort by sex, and what we could see was that this effect was mainly seen in males. So here you have the results for males. Female here, not significant. We also tested additive models versus genotypic models. And the genotypic model is the one that gives the significant results. We followed up with GTEx data. So in many tissues from GTEx, we don't have a lot of individuals. So it's hard to do the stratified analysis in all. But for the ones where we did have enough individuals, we always saw a sex-specific effect. So there was no cases where the interaction effect was significant in both males and females. And here I'm showing two examples of of the highest, the tissues with the highest sample size. So in skin, you see a similar effect compared to what I showed before in Jevadis dataset in the lymphoblastoid cell lines, where we have this increase in expression with A alleles in 477. But with the difference where here, it seems like the effect is reversed in GG individuals. And in females, for the tibial artery tissue, we see completely the opposite, where it actually decreases with each A allele when you're an AA individual, and it increases with each A allele for 7.7 when you're GG. So it was really striking to see those differences depending on the sex. And so just so that you can see that we're not, cherry picking the p-values. Again, we see that these models in males are showing high p-values, but no effect in females. And here, it's the opposite that we are seeing, where the model that the stratified cohort with female is the one giving significant effects. So then we went on to look at the UK Biobank and what association could be seen with the interaction effects. So again, looking at the two SNPs and how whether these interactions were associated with different phenotypes. Again, here we're not in the Peruvian population, we're in the Europeans, white British. And so we see some associations. The p-values are not striking, but some suggestive signals. Notably on these FVC, which is forced vital capacity, and this FEV1, forced expiratory volume in 1 second, so pulmonary phenotypes, so potentially pointing to something related to respiratory phenotypes, potentially linked to high altitude resistance, but this is an hypothesis still. But what you can see is that, again, we see that this effect is mainly driven by females. And then we had very suggestive evidence for cardiovascular disease. We had this interest in that phenotype because of the initial discovery of ADCY9 SNP in a cardiovascular cohort, which again shows suggestive evidence of an interaction effect, but here driven by males. So we dig a bit deeper into this. So that was work done by Marie-Pierre Dubé's group, and in particular Marc-André Legault, who did a bit more into this, testing a three-way interaction. So 7309, 477, and sex, and discovering a significant effect between these. And so in fact, the interpretation is that for the individuals that carry AA genotype at 7309, the association between 477 and coronary artery disease is in the opposite direction in males and in females, such that for the females that are AA, each A allele at 477 is protective for CAD, whereas in males, each additional A allele would increase the probability of CAD, which was interesting because it means that the beneficial effect on the interaction on coronary artery disease would favor males that are AAGG, which is the genotype combination that was seen in excess in the Peruvian, and AAAA in females. So that is very intriguing that we actually recapitulating these genotypes. So in conclusion, we performed population genetics analysis that revealed sex-specific interaction between 7309 and ADCY9 and 477 and CETP. This sex-specific interaction is the first, to our knowledge, to be reported in humans. It has to be, in some sense, if the interpretation of selection is correct, it has to be ongoing selective pressure, because to maintain these LD patterns that are different between males and females, it has to be acting in this generation. So we have different hypotheses for that. So we are wondering if it's a prenatal selection, such that having this combination would favor survival, but differentially in one sex compared to the other. Is it selection at early age in boys? So we're focusing in boys here because the strongest effect was really in males, where we can see that there's an excess of Peruvian males that have this combination. And so is there something related that have higher mortality in boys if you don't have this combination? And also one question that we still have and that we want to investigate is how strong this selection pressure needs to be for us to be able to detect such an effect. And so because there's no other examples, and it has been proposed in theory to exist, but there's no theory necessarily to evaluate this, we think that probably simulations could help in answering that last question. So we found that this association might represent a functional interaction given the results of the expression analysis, again with sex-specific effects, sometimes very confusing, but still with this idea that there's something different between the two sexes. And we are also wondering whether this is a direct or indirect interaction. So we haven't seen, and we've looked in different data sets where we could see that whether there was a kind of a direct interaction between the locus, which with high C data and so on, and it doesn't seem to be the case. So our main hypothesis is probably that there's something in the pathway that is acting so that these interact, but we actually don't know yet. And finally, the big question is, what is the selective pressure on? What phenotype is under selection? So we have some hints about pulmonary phenotypes being associated specifically in women. It's confusing because the male associations seem to be more cardiovascular, pointing towards cardiovascular disease. So, but the pulmonary or respiratory phenotype hypothesis fits quite well with this idea that potentially it comes from Andeans, and there's a link between high altitude that is known to be a selective pressure that is very strong. So still under investigation. And finally, what is the significance for a dalcetrapib pharmacogenomic association? I think at this point, it just means that probably there's something going on between ADCY9 and CETP that might be affecting the response to dalcetrapib, but this will need to be formally tested in next studies or trials. So I would like to finish in thanking the diverse human population genetics data, because this has been enabled, the discovery has been enabled by the fact that we have some diversity cohorts, so cohort from the diversity in genetics today, but still not enough. And then I would like to thank all the people that contributed to this study, and also you for your attention. And I'm happy to take questions.
Thank you very much, Julie. That's really interesting, and it's sort of making our lives more complicated, right? By trying to look at three-way interactions in this field. So, any questions, please? You can put them in the chat, or you can turn on your microphone and speak up. Simon?
Yeah, I was going to say the same thing. Like it's hard enough to pick apart a two-locus interaction. It's very heroic, and trying to tackle with three locus thing. It's super, I mean, I think it's really cool. One thing that you mentioned at the end, which I think is super intriguing, is the selection, which would need to be ongoing and kind of very strong, it seems, right? I mean, so the first question is, did you try to estimate like what kind of selection coefficient would you need, right, for this to be maintained? Because it seems to me that it would need to be huge, right? And
no, absolutely. And again, that's what I meant. I mean, maybe you have ideas, but I'm not sure how really to estimate these selection coefficient when, well, so if we have two loci, fine, but then we have this, you know, additional sex-specific effect. So yes, this is why we are going down the road of potentially trying to simulate some of these. But if you have any other ideas on how to do this, but yes, it's true. And we went into, you know, looking at some register of, you know, male to female death. Well, Isabel did, and we didn't see anything. Like we would have expected, okay, maybe there's, you know, male mortality in early childhood in Peru that is known, but we didn't see that. Or maybe it's just because it's incomplete, right? And we haven't seen, but we are towards, and so, okay, so I don't know if there was a question.
Well, the follow-up question, I guess, was, I mean, I was assuming you looked into that a little bit, but could it be an ancestry by sex kind of bias, right? Where, you know, if you have different proportions of ancestry in males versus females.
So we did check all of that.
And
you
X
could,
versus autosome, yeah. And, you know, I mean, it's just complicated, right?
Right. So X versus autosomes, now we haven't looked really at sex chromosome. That's another hypothesis. So what I was going to say is strong selection happens, but generally this is prenatal, right? Is either sperm biases or, you know, in utero and so on. So my hypothesis is that it's probably there that it's happening. The strongest selection signals are happening there. And then we have all of these potential hypotheses where maybe there's an interaction with the Y chromosome, or having two X's or something, but that would need, anyway. So yes, so that would be kind of the next steps, but would need a kind of a three-way interactions in terms of yeah.
Great, thank you so much.
Jacek, you've turned on your camera. I don't know if that means.
Yeah, I did, and then Simone just asked my question. You know, I had the same idea, and it brought me back to like way back when I wanted to be a population geneticist, and I was looking for linkage disequilibrium between alleles on different chromosomes, until I realized that for this to exist, it would have to be very strong prenatal selection, pretty much. So I gave up, and I never became a population geneticist. But yeah, so, you know, I find that fascinating that it would, and I don't have any solution, just maybe two comments. Is one, you know, if it's not prenatal, it's somehow postnatal, that perhaps, but I don't think there's enough power. You could try stratifying the data by age and seeing if this LD changes in some way. So
we did, and it doesn't, because in the LIMAA cohort we have age, right? So, but the youngest is 18, 16, maybe Isabel remembers, but anyway, there's no pediatric, right, cohort. So we didn't see anything, and when we were, you know, we had this in the paper and saying all these hypotheses, and then the reviewer all said, well, it doesn't mean it's not that if you don't see, you know, the null result, like the fact that we don't see an association, just doesn't help us in some sense. If we had, then it would have been, and Isabel said it's 15. If we had, then it would have meant that, yes, we're depriving, you know, as age increases, if the younger don't have this difference in LD, the oldest have it, then it suggests that potentially it's something related to age. What we think is the solution probably for that specific question would be to have a birth cohort, right? If in boys at birth, it's already, the LD is already seen.
What about it means sperm? What about in sperm, Julie?
Yes, so the problem is the problem with sperm is that you only have one chromosome, so the genotypes. So yes, it could, but we would need to have.
You'd have to look across thousands of them. Yeah.
Yes, and you always need the other parent, right? Because here we have we would need AG enriched, but again, then the maternal chromosomes need to be AG as well. Anyway, but yes, that would be another one.
Another question I had for the geneticists who are here today is what's the mechanism behind the different length of the genome in recombination units, right? The female genome in organs is longer than the male. So is there some hypothesis related to what's going on at recombination that could induce this kind of very long range linkage disequilibrium?
So I don't think we know exactly why. I think so about recombination and female being larger than male in some sense, like but it's not conserved across species. That's one thing that is known, is that sometimes it's females, sometimes it's males that have higher recombination rates. So it's not, and it's thought to be something about proofreading, or at least given that meiosis in female is more problematic, there's more recombination, meaning that there will be some more forces maintaining the oocyte for longer in higher quality if there's more foci in the recombination. But then there's this whole thing about being resolved in gene conversion versus recombination events. Anyway, it's a complicated topic, but in terms of having this differences evolving for having more long-range LD, this is an interesting hypothesis. More long-range LD, for sure, there's longer stretches in males, but in theory, in each generation, it should just be
true
removed, right? So that's what's complicated in some sense.
Are there other questions for Julie? I see that we are at 1 o'clock. All right, then I will say a very big thank you to Julie, and that was a really complicated story to try and tell. So yes, very, very well done, and really thought-provoking. So and generate hopefully some really interesting research for you over the next little while. Thank you so much.
And for everyone, honestly.
Yeah. So we'll have you come back and tell us what's going on in a couple of years.
Thank you very much.
Thank you so much. Bye. Yeah, really interesting.